All right, fellow scientists, so here we go. Today we are going to start talking about biotechnology. So the basic idea behind biotechnology is now that we know how DNA works, now we can manipulate it to serve our purposes. Sorry, that was my best evil scientist laugh. So anyways, let's go ahead and write this down. Biotechnology is manipulating DNA. So changing it, combining it, cutting it, whatever we want to do to it, we can do to it. Manipulating DNA to benefit humans. That's a humans, not a Newmans. Okay, sweet. So we're going to talk about one tool and one process of biotechnology today. So our first tool are called restriction enzymes. And this will be a question on the quiz. What do restriction enzymes do? They cut DNA at specific sequences. They cut DNA at specific sequences. So they're proteins, they're enzymes, you remember the workers in our bodies, in our cells, that do a specific job. They cut DNA at a specific sequence. So our first restriction enzyme we're going to look at is NORO1. NORO1. So what NORO1 does, and I'll draw DNA just so you know that this is a big long strand of DNA that continues, but we're just drawing a little bitty part of it. Right, so what NORO1 does is it'll clamp onto DNA and it'll run down the sequence of DNA and it'll look for these letters, this combination of letters, A, T, T, A. So of course the opposite of that is T, A, T, like that. And what it'll do, once it finds that, it'll stop and it'll cut, just like that. So then what we have is a fragment or a piece of DNA with AT, TA, and then we have our other side, or our other fragment, our other piece of DNA is going to have TA and a little bit longer, AT. So that's NORO1. That's the sequence that NORO1 looks for. By the way, please write these down in your notes because we're going to use these in class when we do a little activity. Another restriction enzyme that we're going to look at is called SMA1. And so SMA1 clamps onto DNA, and it'll run down the DNA sequence. And I made those too close, so I'll just separate them a little bit. Run down the DNA sequence, and it'll look for this sequence. It'll look for C, 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 G, G, G. So, of course, base pairs G, 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 C, C, C. And it'll just, it'll cut right there. So then we have two fragments. All right. So that's one type of restriction enzyme. And notice this restriction enzyme, it just cuts straight. So we call these blunt ends. Blunt ends meaning like they're not sharp. They're not sticky, as we'll see our next two restriction enzymes do. So the next restriction enzyme, and this is probably the most famous restriction enzyme, is EcoR1. EcoR1. So EcoR1, what that does, that will, again, clamp onto DNA, and it'll run down the strand of DNA, and it'll look for this sequence, GAATTC. So CTTAAG, and it'll do something different. It's not going to produce blunt ends. It's still going to cut, but it's not going to produce blunt ends. It'll cut the sugar phosphate backbone right here between this G and this A, and then it'll cut the sugar phosphate backbone right here between this G and this A, and then it'll cut these hydrogen bonds right here, so that what we end up with is mostly a double strand of DNA, but at the very, very end of this double strand of DNA, made it too close again, yeesh. At the very, very end of this double strand of DNA, we're going to have what's called a sticky end. So these nucleotides, TTAA, notice they're not bonded to anything, and so they want to bond to something. So if there's another sequence around close to it that's complementary, then they will bond, they will stick together to the other sequence that's complementary. So then we have AATT here, and then C and G are paired up like that. So that's one restriction enzyme that makes sticky ends. Another restriction enzyme that makes sticky ends, and this is our last one, is Hind3. Hind3. So again, clamps to DNA, runs down DNA, and looks for this sequence, AAGGTT. AAGGTT. And those should be all evenly spaced. TTCCAA. And what it does, it cuts the sugar phosphate backbone right here, cuts the sugar phosphate backbone right here, cuts the hydrogen bonds right here, and then just like with EcoR1, we get two fragments with sticky ends. So let's go ahead and write that down. These are sticky ends. Alright, so I said we were going to look at one tool and one process today. So our first tool is restriction enzymes, and those are examples of what restriction enzymes do. Now, our process, our first process of biotechnology is transformation. So our first process is transformation. And basically what transformation is making a cell produce something that it normally does not. Making a cell produce something it normally does not. So for an example of this, we're going to think about a person with diabetes. A person with diabetes, their pancreas doesn't make enough insulin. And so before we had biotechnology, before we had this process of transformation, they needed insulin from a source. They couldn't make it themselves. And so what doctors, scientists, medical technicians would do is they would go to slaughterhouses for pigs or cows or sheep or whatever, and they would take the pancreases from the carcass of the slaughtered animal, and they would extract the insulin, and then they would give that to humans, to diabetic humans who needed insulin. And so needless to say, there wasn't enough insulin using that method, and so people with diabetes, they just had to try to get along without insulin, which is something that's not the easiest thing in the world to do. But now that we have transformation, we can actually put human DNA into bacteria, and we can have that bacteria make insulin, which is something that it normally does not. So it's a relatively simple, straightforward process if we just look at the basics of it. So we're going to take some human DNA, and human DNA, remember, chromosomes are linear. So we're going to cut the human DNA with EcoR1, which, remember, makes sticky ends. Cut with EcoR1. And because we know the sequence of this gene, of this DNA, we know that we are cutting out the gene for insulin. The gene that makes the mRNA, that makes the protein insulin. So that's our human DNA. Then we take a piece of bacterial DNA. And remember, bacterial DNA is circular. And we are going to cut that with EcoR1, with the exact same restriction enzyme. Right? What that allows us to do is we're going to have sticky ends that can pair up with each other. So we're going to have this cut piece of bacterial DNA, and if we put it together with our piece of human DNA that makes insulin, now we're going to have a circular piece of DNA that's mainly bacterial DNA, but it has this little gene that makes insulin, the human DNA. So then what we're going to do is we're going to take that, and we are going to put that into a bacteria. So we put this small little circular piece of DNA into a bacteria, and then lo and behold, because this bacteria has human DNA for insulin in it, now our bacteria makes something that it didn't before. It makes insulin. So then we can grow a bunch of these bacteria and we can harvest the insulin, and now we have all the insulin that we want, that we need for people with diabetes. So as cool as that is, that's not the coolest thing that we can do with transformation. Consider this jellyfish. This jellyfish produces a protein that glows green when it's exposed to ultraviolet light. Pretty sweet, huh? What if we put that into other things, other organisms, other cells? And we have, in fact. We can put it into bacteria. In fact, this is a lab that we do in AP biology. We take normal E. coli bacteria and we put the gene that makes the protein that glows green into these bacteria, and then we shine ultraviolet light on them, and lo and behold, we have glowing bacteria. Can we do this with anything else? Yes. You can have your very own glowing fish. Now, fish don't normally glow. At least most fish don't normally glow. But if we take genes from different fish or different corals that normally glow and we put them in the fish when they're a one-celled embryo, then every cell of that fish is going to have that glowing gene, and you can actually go to the pet store. These are called glowfish, and you can get your very own glowfish. Pretty sweet, huh? We've also done this with mice. We've also done this with rabbits, and lo and behold, chimpanzee. Right? Now, of course, it would be unethical to do this as a human, to a human, because you would have to do it when they were a one-celled embryo, and yeah, plus it's kind of scientifically difficult, let's say, to do that. So, what's the whole point? What's the benefit of having glowing green animals? Well, it's really sweet. We can attach this green fluorescent protein to a different protein, and we can use a protein that we can see to identify where a protein that we can't see is active. So we can study embryos and we can look at what proteins are active and what proteins are not active in embryos. And here's an example. There's a certain protein that this green fluorescent protein is attached to that is made whenever the other protein is made, and it's only active in this certain part of the embryo. So that's a huge tool in biomedical research, and it's increasing and advancing our knowledge of how embryos develop and how DNA works and how the body works. So anyways, those were our first little forays, our first topics into biotechnology. First tool, restriction enzyme. First process, transformation. By the way, transformation will also be on the quiz. What transformation is, the definition of transformation. All right, so hope you enjoyed, and I will see you in the next video.